Okay, we're going to try something a bit different for this last bit. One of the things that we can do now that we're doing blended learning format, where we're doing, or remote learning format, I should say, as well, now that we're doing studying at home and doing it on our computers, is that you might be able to run alongside me an analysis using various different pieces of software, which we wouldn't be able to do if we were doing it in a lecture theatre, or a computer lab would be possible, but that's a bit more tricky to embed in a lecture unit. Anyway, what we're going to try and do is that if you can, alongside me, try and pause this video to try and do various different stages to run an analysis of your own, a phylogenetic analysis at home on your own computer. We're going to follow a example using coronavirus sequence data to try and understand the origins of the human coronavirus in comparison to a zoonosis. Where did it come from? To do that, we're going to use a Phylogeny Reconstruction Workshop put together by Jonathan Dury at the University of Durham, who's put this together and asked other people to share it. I think it's quite a good exercise, so we're going to use that as well. So here he's put together some sequences and a workshop code about how to do it. So in order to do this exercise at home, the first thing you might want to do is download RStudio. This is a free piece of software. You may have come across R first of all in your training earlier stages. So have a look, download that, and when you're ready, unpause the video and have a look. So once you've done that, this is what R will look like. Opening up RStudio, you'll start down here with your command line. Over here is some things that I've copied and pasted from the workshop. And over here is where we'll see the plots to visualize it. Okay. So one of the things that you'll have done is I've made the sequences available on the Blackboard web page. Download that and put it in a folder on your computer. That's just the sequence information. More about that in a minute. First of all, we need to get ourselves up and running. So what we're going to do is then set the working directory. So I'm going to choose the directory here. And I think I've put it in my Dropbox folder under teaching, under BIOL21232, and there's the file, COVID region B FASTA. That's the sequence information. So once I've done that, I've opened it. And now my R is going to operate within that folder. I'm going to load up these packages that will help it to do what I need to do. So you could just do this by copying and pasting from the workshop information. This may take a while. Okay, I've installed those packages. That took a little while. You may be asked as you're doing it to prompt to say which ones you want to do, whether you want to update things. Just choose your yes, no's or your alls, and hopefully you're ready to go. So for the first part, we're going to load in the DNA sequences. As part of this workshop, what's been put together is a series of sequence data from a number of different genes. These are accessed from using the GenBank, an online repository of lots of different genetic information. Here are some of interest. That there are lots of bat coronaviruses. Here's the human one that's going to be part of one of the first earlier ones identified in 2019. And here is the earlier one from the SARS outbreak from the 2003 outbreak, which is we may be less familiar with. There's information about how to access them here. So now that we've seen where we can load them, we can if we just copy and paste this command from this worksheet into R, this is now created an object of the sequences, right? We now have sequence information for over 5,000 base pairs of DNA base pairs of these, I can't remember how many or so, 14 or so taxa, okay? But we have to think about the sequence alignment to try and establish that we're working with homologous points. So at the moment, this is what the data look like, right? We're dealing with a situation where it's all messy. There will be insertions and deletions, which means that we haven't aligned it and we don't necessarily know which point in those DNA sequences are homologous. So the first thing we need to do is undergo a sequence alignment. And that's what we're going to do here with this particular method using the muscle method and the MSA. So we're going to take that and align it. That may take a moment for the computer to figure out how to do that. Okay, I had to pause the recording to give that enough time to catch up, but that's now finished. So let's have a look at the results and print them. So this is going to print those results and show the whole lot. Okay, so there we go. There's the sequences for nearly 6,000 base pairs for each one. Now looking nice and neat and tidy. That's obviously not a great way to visualize it because that's page after page after page of sequence information. So let's try and trim it off and have a little look at it in a more tidy picture. Okay, so that's taking off some of the edges, and then we're going to try and have a look at it as well afterwards. If you wanted to, you can have a look into the further detailed explanation in the workshop and see what each of these commands actually means. But let's create a nice image. There we go. And now this is our updated image of the sequence alignment. Now we can see that that's more tidy than the original unaligned version. Over here, we can see that now we have like stripes indicating that those sequences have been aligned and that we have some homologous points along that DNA sequence, or at least we've minimized it in order to make the most sense across those taxa. Okay, now we have our sequences in, we've loaded them up, we've got them all aligned and nice and tidy. The next thing to do is to think about how to actually create a phylogeny from that. So doing a parsimony analysis in R is a bit tricky, so we're going to start with a UPGMA, which is roughly similar but not really as good, and we're going to use that as a quick and dirty approach to try and identify the similarities between these sequences and use that to create a tree. So that's what we're going to do here. This first command here is just doing converting it into an object in order to allow it to do an UPGMA, and then we're going to say UPGMA tree that's going to create something called UPGMA tree, doing UPGMA on our sequences. That's the important stage here for this tree building. Now that's quite quick and dirty, and now we can have a quick look at it. There it is. Now the default is quite small on the text, so let's increase that size. The CEX is what that's doing that. So let's make that, that's a bit messy. Let's try again. Need to be able to read the text and see the tree. Okay, we still can't see that very well, but never mind, that'll have to do for now. Maybe let's try the first one. Okay, I don't know if you can read that on your YouTube screen very well. So what we're looking at here is the alignments and the sequences. So we can see the similarities and differences. But in the previous video, we talked about being rooted and unrooted. And by default, this is unrooted. So we may want, in order to make sense of this in an evolutionary perspective about what came from where, we want to make some sort of decision about which taxa we're going to root this on. So this is where we're going to root it on these particular strains, which we might understand as having a more longer evolutionary lineage or being more distant from the others. So this helps us to visualize the tree that we're looking at here. Let's try and make that a little bit bigger in terms of the text so we can see it. Okay, so in this one we've now got a bit of a tree. Let's try a different thing. Can we zoom in on that and see it in a bigger fashion? There we go. So this is our quick and dirty parsimony style UPGMA analysis, which isn't really parsimony. And we can see here that the important one, the most recent outbreak of the coronavirus, so this Wuhan virus isolate from 2019, is here, right? And that is closely related to this bat coronavirus, which is also closely related to a bat SARS-like coronavirus. So they're in a little family there, nested within pangolins. There's also another area of bats over here with where the other outbreak of human coronavirus has taken place. So we can see two separate origins for these coronaviruses, these SARS-like coronaviruses that have infected humans, and they both, in two instances, come from two different groups of bats, one more closely related to pangolins. And indeed, if we were to treat this as a phylogeny in an evolutionary sense, then we would infer that actually what's happened here is that it's been in pangolins and then jumped into bats, because this is the pangolin ancestral lineage, then along bats and then into humans. However, as we talked about, this is not an evolutionarily informed model. This is just based on the sequence similarity, in much the way that parsimony is. It doesn't take into account any understanding about how things evolve in a molecular level. So in order to do that, we need to move on to a molecular approach, a maximum likelihood approach. And that's what we're going to do with the next stage here. So this is where we're going to take that tree and put it in some probability models where we can understand what's going on and try and extract a maximum likelihood, which is what we're doing here. In this one, when we talked about the different models we might use, we're using a GTR model. This is has some parameters. It's a commonly used model which allows 6 rates of evolutionary change depending upon the types of nucleotide changes taking place in the evolutionary model. So that's going to take a little time to have a little think about it. It's undergoing quite a lot of different algorithms to try and think about this in different ways, to try and think about making it the most probable tree. So now we're not just thinking about which makes the sense on the basis of the broad similarities and differences. Now we're asking what makes sense in light of what we understand of how evolution would work. Now, at this stage, we can have a little output and have a look at what the transitions and transversions are, about the probabilities of moving from one to the other, right? From the A's to the C's to the G's to the T's. That's what we're using. And it tells us which ones are most common and which ones are least common in terms of the A's, the G's, and the C's and the T's. And that could make a difference in the way that these evolutionary rates work. So let's get this phylogeny up and running then. Let's extract the tree from this and then plot it. Rerouting it like we did before. It's hard to visualize because of the small letters. Let's try and do it without that. In fact, let's just open it without any of that and see if we can visualize it properly. Here we go. We can't fit in the sequences. One moment, I'll try and see if I can tidy that up. Okay, this is the maximum likelihood tree. So once again, we see a similar sort of picture in that the Wuhan human coronavirus, CO2, is here, inside this bat family, close to the pangolins, and then there's a separate one over here for the earlier one. Now this time the phylogeny looks slightly different. The human coronavirus that infected humans here in 2003 is now closer to the root of this particular one, rather than the other one. It was nested quite firmly within bats. Again, that has different implications for the evolutionary history of these viruses. So what's interesting here is that we have now reconstructed our own phylogenies using different kinds of inference methods and compared them in terms of the similarities and differences. Overall, they're showing roughly the same amount of evolutionary patterns, and that we can relate the coronavirus outbreaks in humans in both the 2019 variant and the 2003 variant in different parts of the family of animal coronaviruses, and tell us different things about the zoonotic events. There are different things that we could do. We could do a formal parsimony analysis. We could do bootstrap analysis to try and estimate the amount of support on that. And indeed, that's part of this exercise. If you want to try that yourself, you can see which parts of this tree are supported by stronger or weaker evolutionary signals. In fact, there are all sorts of different things that you could do. But I thought it would be nice for you to be able to have a play around and your own time. Now, don't worry if you weren't able to get your programs to work or are to work on your computer. You can just follow along with the basic principles here, but it might be worth, if you can, at least have a try to see if you can create your own phylogeny. Because then, quite frankly, you're away. You are an evolutionary biologist able to do important and useful things, and it will help us think about the intended learning objective of this course and this unit. During the live session, we're going to have another go at creating a morphological phylogeny live in the session, and then you will be fully qualified evolutionary people. Thanks.